Hello, guys. This is the second lecture on chapter 3, amino acids, peptides, and proteins. Just 1 second. Мам, потише сделай. Мам. Потише сделай. I'm sorry, I had to ask my mom to turn down the volume on her computer. So this is from September 2nd. Amino acid, peptides, and proteins. So second lecture on chapter 3. So I showed you amino acids last time. Showed you a bunch of amino acids, and we grouped them into 5 categories. We grouped them into five categories. I need to share my screen. Just 1 second. Share my screen. There we go. Share my screen. All right. Five categories. Let me get this pen. Right, so there will be, remember, non-polar aliphatic, which means non-aromatic, right? But non-polar. Aromatic ones, 3 of them, polar and charged, and then ionized, positively charged and negatively charged. So we went over these briefly, and maybe not so briefly, but we talked about various side chains. And remember what are some key points that we should have taken from the lecture on Monday, on August 31st. So glycine, remember, so each amino acid, there is a, in 19 out of 20 amino acids, there's a tetrahedral carbon, right? 4 different substituents, carbon is asymmetric, so the molecules are chiral, with the exception of glycine. In glycine, 2 hydrogens are the same, so the carbon is not asymmetric, and so glycine is achiral. It's the simplest amino acid, achiral. So then we add alanine, add methyl group, result in slightly more complex alanine. Now also keep in mind that there is 1 amino acid out of 20 where the nitrogen is secondary, right? So you can see there is a bond towards this carbon and there is a bond towards this carbon, right? Compared with alanine, for example, where there's only 1 bond towards the carbon, or glycine, one bond towards the carbon. So proline is the only amino acid out of 20 where you have a secondary nitrogen, a secondary amine. Even though the carbon is still chiral, this alpha carbon, right? So we refer to this as alpha, 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 and so on and so forth. Now we increase the group R, go to valine, leucine, isoleucine. Remember, difference between leucine and isoleucine is the position of the methyl group, very important. Now methionine, I kind of told you that even though sulfur is a heteroatom, right? But it's not as electronegative, let's say, as oxygen is, right? So methionine is considered kind of halfway between polar and non-polar. So, but we put it into a non-polar category. Now there are 3 aromatic groups. So phenylalanine, tyrosine, and tryptophan. The difference between phenylalanine and tyrosine is the presence of this OH, right? Which makes it capable of hydrogen bonding. And tryptophan, remember, there's this beautiful heterocycle known as indole. indole. So, and remember these especially tyrosine and tryptophan, they absorb UV light, which makes proteins absorb UV light, and that's due to these two amino acids, right? Tyrosine and tryptophan. Aromatic systems in general absorb UV light. They have pretty high extinction coefficient. Then we have a polar but uncharged, so serine and cysteine. Now cysteine, remember, compared with methionine, now we have an SH. It's a sulfur, but it's capable of hydrogen bonding, and sulfur is quite nucleophilic. So we put cysteine into polar uncharged category. Now threonine and serine both have OHs. The difference is this additional methyl group. Now asparagine and glutamine are amino acids which have amides in their side chains. And remember, amides are quite polar, right? Amides are quite polar. And the reason for that is because of the resonance structure that you can draw. You can draw a nice resonance structure. Like so. There's a lone pair comes in here, goes there. So amides are quite polar, even though they are uncharged. Plus, minus. Let me start the timer. I would say we've been talking for maybe about 7 to 10 minutes, right? So 1 hour, 10 minutes left. One hour, 10 minutes. All right. So, and what's remaining are charged groups, right? So there's positively charged groups, lysine, arginine, and histidine. So now there is a small mistake here. We were gonna put a proton on this nitrogen, and that will make it charged. And arginine is charged, and lysine is charged. And remember that arginine is more basic than lysine, and the reason for that is because the lone pairs on the nitrogen, they can come in and donate electrons towards this nitrogen. And the same thing here. Same thing here. Like so. All right. So lysine and arginine and histidine. And finally, the fifth group, negatively charged amino acids. So these are carboxylic acids, aspartate, and glutamate. What we'll find out is that the pKa values of these are quite low. So at physiological pH of 7, they will be deprotonated. Now, just to remind you, physiological pH is around 7 to 7.4, depending on where you are in a biological system. 7.4. So if your pKa of your carboxylic acid is 7, let me put if here. if pKa is 7, that means 50%, oops, get rid of this. That means 50% of your carboxylic acid is deprotonated. I know I already said it before, but I will say it again because it's very important. Deprotonated. It's a very important piece of information that you use to predict whether your carboxylic acid is protonated or deprotonated. So then if your pH is below 7 to 7.5, right? Then your carboxylic acid will be protonated. If your pH is above 7, above 7, so in other words, if pH is higher than 7, it will be deprotonated. If the pH is below 7, it will be protonated. And the degree of protonation depends, so these are orders of magnitude, right? So let's say if the pH is lower than pKa by 1 unit, that's 10 times, right? So let's say pKa is 7, pH is 5, then the ratio will be 100 to 1. So 2 units, 2 orders of magnitude, right? It's 100 to 1. So for each molecule which is protonated, deprotonated, there will be 100 which are protonated, and vice versa. Same for glutamate. All right, so I want to spend a little bit of time talking about these amino acids which are not very common, not parts of proteins. Let's see, I have more. Yep, I have more here. Well, they are, some of them are parts of proteins, but not very common. So hydroxyproline and hydroxylysine are components of a protein known as collagen. So we will learn that these hydroxyl groups impart hydrogen bonding properties within the three-dimensional structure of collagen and make it very strong. And so, so the way these are introduced into the protein is through what's known as a post-translational modification. Translational. So translation is on a ribosome, right? So it's where proteins are synthesized. Post-translational modification. So there'll be enzymes which will come in and oxidize this position of proline to introduce the hydroxy group. Modification. And that's the most common way, except with some exceptions. I'll mention what they are in a moment. Modification. Now, N-methyl lysine. Now, that's a very important for genetic regulation, right? So, so lysine is an amino acid which is very common with the types of proteins known as histones. Histones, which regulate gene expression. regulate gene expression. And so one way they do that is through methylation of their terminal nitrogen on the lysine, right? So the methylation status of this nitrogen is very important. So histones carry this kind of modification. Now carboxyglutamate. Now this is a calcium binder. So we introduced another chelating group, right? Introduced another chelating group. And metals love chelating groups. So actually, let's see. So found in blood clotting protein prothrombin and other proteins that bind calcium as part of their biological function. Now, what's this desmosine? Very interesting amino acid. You can see actually there's a pyridine ring in the middle, and then there are these amino acid residues on the termini of this interesting molecule. So desmosine is part of the protein known as elastin. Elastin is an important enzyme in glycolytic pathway, and we mentioned that earlier in this class. Elastin. Now, selenocysteine. Now, very unusual to see selenium in the biological system. What part of the reason is because selenium is actually toxic. So this is not present in large quantities, but it's an analog of serine. If you think about, if you replace selenium by sulfur, oh sorry, not serine, cysteine. Replace selenium by sulfur, what do you get? You get cysteine. Plus minus. So this will be cysteine, right? And so, so this participates in reactions where selenium has more beneficial properties. So just think about it. Selenium in a periodic table is right underneath sulfur, right? So it's heavier, it's more nucleophilic. And so wherever such properties are needed, that's where you will find selenocysteine, but not very common. And pyrrolysine, okay, so they're saying in the text, it's a methanogenic bacteria. Methanogenic bacteria. So it's pyrrolysine is part of the microbiological world. Microbiological world. Okay, and a few more important amino acids. Then we have 3 amino acids here: phosphoserine, phosphothreonine, and phosphotyrosine. Now, these are crucial in biological signal transduction. Signal transduction. Signal transduction. So how is this signal transferred within the cell or from outside of the cell? So let's say you have a hormone, right? Arriving, let's say insulin, right? So let's say you have an elevated blood sugar, and insulin is produced by the pancreas, right? Insulin is produced by the pancreas, goes into the bloodstream, and then what does it do? It binds to the surface of cells of different tissues and tells these cells, listen, there's increased sugar in the blood. I want you to express receptors known as, for example, as GLUTs. And those receptors will bind to glucose in the blood. So GLUTs means stands for glucose transfer, right? So so these will bind to glucose in the blood and allow for the glucose to get inside the cell. So, but how does hormone like insulin, how does it tell the cell that that it's time to act, that it's time to produce these GLUT transporters? And so this comes from what's known as signal transduction pathway, right? So basically biological systems get activated, and many proteins that, let's say, have tyrosine residue will undergo phosphorylation. So remember the enzymes which phosphorylate hydroxy groups in biological systems known as kinases. Kinases put on phosphate. And the enzymes which work in the opposite direction are phosphatases. So these enzymes remove the phosphate. Remove the phosphates. And so these results in what's known as signal transduction cascades, right? So let's say insulin binds to insulin receptor on the surface of the cell. Insulin receptor phosphorylates itself. Very interesting. Some parts of the receptor serve as a kinase. Another part of the receptor serves as a tyrosine residue, which will undergo phosphorylation by this kinase. Once that is phosphorylated, then another molecule will come in, will recognize this phosphate, and it'll undergo activation, and so on and so forth. So these amino acids will undergo phosphorylation reversibly, right? Depending on where they are, what they do, and at what stage of the signal transduction pathway they are found. So all three of these, phosphoserine, phosphothreonine, and phosphotyrosine, play a role. And no wonder that actually many of these pathways have been hijacked by the cancer cells, right? So the signal for cell division also works through these phosphotyrosine signal and transduction pathways. And so basically, if there's a growth factor which arrives to the surface of the cell, basically it keeps telling cell divide, 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 divide. And so these tyrosines will undergo phosphorylation, and then the signal will be taken into the nucleus, where various transcription factors will initiate cell division, or will produce proteins which will initiate cell division. So and in many cancer cells, these pathways have some kind of abnormalities in them. And so, for example, various controls do not work, right? So, for example, a particular kinase, which has some kind of a way to be regulated, has undergone mutation. And so that mutation will prevent this kinase from slowing down. And so the kinase will be constantly putting on these phosphate groups onto the tyrosine residues of various receptor proteins and protein substrates, and the cell will be in a constant process of cell division. There are actually a number of drugs on the market right now which work by inhibiting these kinases. And so, for example, we will find out later in this course that these phosphates come from ATP, adenosine triphosphate. And so many of these drugs occupy the binding site for ATP at the active site of these kinases. And by doing so, prevent the ATP from binding at the active site. All right. I think we've talked quite a bit already. I think this should be good enough. I think this will be a good introduction for us in terms of amino acids which can be modified, right? Amino acids which play smaller role. Well, you can't really say that the phosphorylation is a smaller role. It's just that they're not part of the 20 amino acid group that constitute proteins. All right, so let's move on. Let's talk about ionization of amino acids. So amino acids have amino groups and have carboxylic groups. So carboxylic group obviously has it has protons that it can give off, right? So carboxylic group can lose a proton to form a carboxylate and protons. Now, but also the amino group, right? Even though by itself it doesn't have a proton, but at high pH value, you can protonate that, and it becomes an acid, right? It can lose this proton. So amino acids actually have there are two possibilities for it to have ionizable protons, one through carboxylic acid and one through the ammonium ion of the amino group. Okay, so let's look at how this will occur. So these are different amino acids arranged in terms of, well, not just amino acids, various compounds, arranged in the order of their pKa values. So this is the acetic acid. So we've talked about acetic acid before in organic chemistry, and we know the pKa it's a weak acid. pKa is around 4.8. Okay. So at this pKa, acetic acid will lose a proton and form acetate ion. What we should notice as the first thing that might attract our attention is that glycine as an amino acid will lose its proton, but its pKa has shifted from 4.8 to 2.34, right? So if you ask yourself that question, why that's the case, probably the difference is the presence of this protonated amino group. And so what happens is that, remember, you can stabilize a negative charge by pulling electrons away from it, right? So if you have O minus, so the protonated nitrogen, which is positively charged, is a strong electron withdrawing group. So it'll pull electrons, remember, through inductive effect, it'll pull electrons away from the carboxyl group and away from the negative charge. And by doing so, it will stabilize the negative charge. So the conclusion is that carboxyl group in amino acids is much more acidic than in carboxylic acids. Well, much more, basically, what are we talking about? Well, in this case, it's over 100 times, right? So remember, again, 1 pKa unit is 10 times. So if you compare 4.8 and 234, here we have more than 2 orders of magnitude difference. All right. So on the other end of the spectrum, so if we go to very high pH values, so methylamine has a pKa of 10.6. So the protonated methylamine, the conjugate acid, has a pKa of 10.6. Now, if you compare this with glycine, then the pKa drops to 9.6. And I think it's the same idea. It's that the carboxylic group will move electrons away from the nitrogen. And if you had a protonated nitrogen, it would destabilize it, right? It would destabilize it, and therefore it will make the protonated amino group more acidic compared with the methylamine. And you can see here, it's 10 times, right? It's 10 times. So in between, so it's possible that you, because the pKa values of glycine in terms of carboxylic acid is 2.34, the pKa in terms of the amino group is 9.6, anywhere in between, you will have a nice ion known as Zwitter ion. Zwitter from the German word double. Zwitter ion, where the ammonia group is protonated, but the carboxyl group is already deprotonated, or vice versa. The carboxyl is deprotonated, at the same time you can have your amino group protonated, right? So in other words, you would think that that it's more reasonable just to have this proton jump back from the amino group to carboxylic group, but that's not the case because of the difference in pKa values. COH. No. No way. Zwitterion is the way to go. All right. I think this is what I want to say on the slide. And the reason for the formation of the zwitterion. So at low pH, the amino acid exists in positively charged form cation, high pH, negatively charged anion. In between, it will be a zwitterion in which the single molecule has both positive and a negative charge. So let's see, and let's look at the same concept using titration. So let's say I have not added any hydroxide, right? So, and I fully protonated my glycine. So my carboxyl group is protonated, my ammonium group is fully protonated, and so I'm starting at a very, very low pH, somewhere there. And then I'm gonna add about half of the equivalents of hydroxide ion. What's happening? I will be deprotonating carboxylic acid because the pKa is so low, right? It's 2.3. pKa is 2.3. I'm going to start deprotonating, and at some pH value here, this pH will equal my first pKa. I'm going to designate my first pKa as pK1. pK1 because there are two pKas. So pKa1, and once I reach the 2.34, I will have deprotonated half of the carboxylic acids. Okay, so 1/2 deprotonated. And then I'm going to continue adding OH minus, and I'm going to continue deprotonating this carboxylic acid until all of it has deprotonated, right? And so this is what's known as isoelectric point. Isoelectric point, where the amount of my carboxylates equals the amount of my ammonia, or protonated amino group. So here it's I've reached the point of equivalence, and also known as isoelectric points. And this is, I have kind of complete zwitterion. Okay, but I'm going to just continue adding OH minus. I'm going to continue adding OH minus, and I'm going to reach a point at which I will have deprotonated half of the NH3 plus. I'm halfway between NH3 plus and NH2, and I've just reached second pKa, the second pKa value, which is pKa2, right? So this is the pH at which half of the amino groups are deprotonated. And if I can continue adding OH, then I will deprotonate everybody. Everybody is gone, and I have a very high pH value in my solution. So zwitterions predominate the pH values between the pKa values of the amino and carboxyl group, right? So if we go back, so it's between here and there. This is where I'm gonna have my zwitterions. Zwitter. ion zwitterine. Okay, and it's very easy to, if you have a pKa1 and pKa2, then it's very easy to find the isoelectric point or the equivalence point. Basically, it's the mean of the two, right? And at this point, the net charge is 0, right? And what does this mean in terms of physical properties? Well, first of all, is amino acid is least soluble in water, right? Basically, I lose any kind of charge. Well, I have two charges, but they compensate each other, right? So as a zwitterine form, amino acid is the least soluble in water. And also, if I apply external magnetic field, my amino acid will not migrate, again, because it doesn't have a full charge to do so. Okay, so because I have a carboxylic group there, and I have the ammonium group, right, protonated amino group, then actually amino acids can serve as buffers, and it will actually can serve as buffers in two different regions of the pH, right? So it can serve as a buffer around 2.34, and the reason why it can do that is because of the carboxyl group. And I can also utilize the buffering capacity around the amino group, which is around 9.6. Because again, buffers prevent change in pH close to the pKa, so glycine can act as a buffer in two pH regions. So let's look at that, right? So you can see it's a pretty flat area here, right? So even though I have added I have added almost one full equivalent of OH minus, but my pH has only changed by how much? By less than 2 units. Even though I added so much base, one full equivalent. So the same thing here, right? I will add a lot of pH, sorry, a lot of OH minus, but my pH will change by 2 units only. So these are buffer regions. So amino acids can be used as buffers, and even though there are better buffers, but sometimes it's useful to have these kind of buffering capacities in two different regions of the pH scale. Okay. Now, what happens? So glycine was the simplest case, right? So what happens if our amino acid by itself has additional ionizable group? So what happens if the side chain can undergo protonation as well? So let's look at histidine. It's one of those. So here we have when in a fully protonated state, carboxylic acid is protonated, amino group is protonated, and the nitrogen of the histidine is protonated. And I have a double positive charge. And so this will occur at very low pH. Then once I start adding OH minus, okay, at a certain, I'm gonna start deprotonating carboxylic acid. I'm gonna start deprotonating carboxylic acid. And after a certain period, after I've added 0.5 equivalents of OH minus, I have deprotonated about half of my carboxylic acid groups. And so that will be the first pKa of 1.82. And you can see actually in histidine, this is even smaller than in glycine. Remember, if we go back to glycine, this was 2.34. But in histidine, it's even lower. It's 1.82. And the reason for that is, again, because of all these electron inductive effects, right? So this nitrogen will pull electrons. This will pull electrons. That will pull electrons. So both positive charges will pull electrons towards themselves, which will make the carboxyl group much more acidic and will facilitate deprotonation. And so the first pK1 is even smaller, 1.81, sorry, 1.82. So once I've added one full equivalent of OH, I'm somewhere here, right? So at pH of around 4, I will have completely deprotonated the carboxylic group. And the charge that my molecule has gained is +1. It's +1, and the reason for that is because I have 2 positive charges and 1 negative charge. The sum of those is +1. Anyway, I keep adding my OH minus, and I keep adding my OH minus, and then at the second pKa, now we're going to call this pKr, and R stands for side chain. So in other words, this pKa arises due to the ionizability of the side chain. So here the pKa is about 6.0. And at this pKa of 6.0, I will have lost half of my protons from the histidine. Half of my protons from the histidine I've lost, and that happens at pH of 6.0. And I keep continue adding OH minus, and here I've reached equivalence point at which histidine is fully deprotonated, carboxylate is fully deprotonated, and all I have is just the primary amino group, which is still fully protonated, but the charges are 0, right? And that's why it's equivalence point, because my molecule is neutral. So my molecule is as a Zwitterion. My molecule is a Zwitterion. And then I just go crazy. I keep adding more and more OH minus. I reach the second pK2 point at which half of my amino group has undergone deprotonation. And eventually, after I've added 3 equivalents of OH minus, I have reached a point where everybody is deprotonated. No more protons left in the molecule. So I've removed 3 protons from the molecule. And the molecule gains a full negative charge because of the carboxylate, -1. So this is a titration curve for histidine. So you can set up this kind of titration curve for any amino acid. Just basically what you need to know is the pKa values of each of these. So if you see this on the test, those will be given to you, right? So I'll give you pKa, the first one of 1.82 for carboxylic acid. I'll give you one for histidine, 6.0. And I'll give you one for that, 9.17, right? So how to calculate the PI when the side chain is ionizable? Identify species that carries a net zero charge. That's it, right? That's the species. Identify the pKa value that defines the acid strength of this Zwitter ion. And that's that. Identify the pKa value that defines the base strength of this Zwitter ion. That's pK2. That. And then take the average of these two values. What's the what's the PI of histidine? Well, this was on the previous one, right? So so the PI will be the average of pKr and pK2. Okay. All right. So, so I think that's, we have covered the amino acid material in great detail. Let's move on now to peptides and proteins, which amino acids form. Okay. So obviously, we already know that the way that it's going to form is by condensation reaction, right? So you have an OH from the carboxylic acid, you have a hydrogen from an amino group, which will combine to form water and combine to form an amide. Combine to form an amide. So in this case, we just formed a dipeptide. Dipeptide. So di stands for there are two amino acids in this peptide. Dipeptide. And in general, we're going to refer to molecules which have molecular weights of less than 10 kilodaltons as peptides. 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 Now, if more than 10 kilodaltons, now, by the way, Dalton is the atomic mass unit. If you are not familiar with this nomenclature. Kilodaltons. Kilodaltons. Oops. I'm getting tired. Eraser. Let me erase this whole thing. If the molecular weight is more than 10 kilodaltons, we will refer to these as proteins. Now, sometimes you will see this term polypeptides, which I've never been able to understand what that actually means. Because there is peptide and there's a protein. Why do we need another one? Polypeptide. What is polypeptide? If you guys have an explanation of polypeptide for me, please throw it in a discussion on Canvas. So amide bonds are pretty strong. Amide bonds are pretty strong. Now compare. So there's a reason why nature has chosen amide bonds. So compare amide with let's say an ester. So let's say we're going to hydrolyze this. And we're going to hydrolyze that. And amide's hydrolysis is extremely slow. And ester is fast. And the reason for that is I drew something for you earlier. So you get your carboxylic acid back. Carboxylic acid back. Plus NH2 R prime. And the reason for that is the resonance. Resonance, remember something that you did not like to draw in organic chemistry? But it's the key to explaining a lot of things in chemistry and biology. Reason. So the lone pair on the nitrogen will donate into the carbon-carbon bond, and these electrons will move towards the oxygen. And here we have positive charge. Here we have positive charge. So there's a reason why nature chose amide bonds, highly stable. And just imagine if our proteins were made out of esters, right? So they will not survive basic biological processes and degrade upon the first opportunity. Now, peptide ends are not the same. So numbering and naming starts from the amino terminus, right? So, so here we have what, 1, 2, 3, 4, 5. This is pentapeptide, right? And so we're gonna name this pentapeptide starting from the left, right? We're gonna put them in a group on the left and start naming this, right? So now this is naming using full amino acid names. So serine, glycine, tyrosine, alanine, leucine, using the 3-letter code abbreviation, serine, glycine, tyrosine, alanine, leucine. And for longer peptide-like proteins, the 1-letter code can be used, right? So we're gonna be using that. And there'll be, so there's a reason why we would like to learn this so that we can utilize it for longer peptides. So now peptides by themselves. So before we move to proteins, let's quickly talk about peptides. Now insulin, even though it's, what's the molecular weight of insulin? Is it, does it fall within 10,000? Let's see. Insulin. Because I think insulin can be referred to as protein. Molecular weight. Oh yeah, it's 6,000 Daltons, so it's under 10,000. So insulin can be considered as a peptide. And we already mentioned insulin in this class a couple of times, right? So secreted by pancreas and signals to various cells that the sugar or glucose in the blood is elevated. Oxytocin is a hormone which causes uterine contractions, right? Sex peptides important for insect mating. Now there's various antibiotics, toxins. So a lot of toxins produced in the microbiological world are actually peptides, right? So chlorotoxin by scorpions. Variety of biological functions, but obviously they're not capable of doing things that proteins do, just because they're just too short, right? Just too short. Just think about it. So even though you may need just, let's say you want to catalyze a reaction, right? And you have an active site. So let's say at this active site, you put the, I don't know, 100 amino acids or something like that. But that's not enough to catalyze a reaction. Even though you may make all sorts of necessary bonds with the substrate. But what about regulation, right? So this active site just sits there and catalyzes the reaction every time the substrate goes by? No, there must be possibility for regulation, right? So there must be other small molecules or other proteins which will interact with other parts of this peptide or protein to tell it when to shut off, to tell it when to work, when not to work, right? So there's a reason why proteins are so large. And so that's why peptides, their functions are somewhat limited or significantly limited compared with proteins. But they're still important in, you can see, still have a variety of functions. Many of them are cofactors, many of them are coenzymes, prosthetic groups, other modifications, and on and on and on and on. I think we're gonna stop here. We have a few minutes left, but we're gonna stop here. And have all guys have a good break, right? Have a good break, and find some time to do some work. And I would like to see some more questions and more answers too. So I know that you guys keep asking questions, but you keep asking questions from me, right? So I would like you to actually talk to each other as well. So, all right, have a good break, and I'll see you all on Wednesday.